We will continue our talk about protists, where we go through all the different lineages and some examples of each. I hope you walk away from this with an idea of how spectacular the microbiotic world is. Molecular data, which is, to me, the way you should organize things. Molecular data places what we call protists into every single supergroup out there. They're everywhere. They're part of everything. Supergroups are a level that fall right underneath the domains, taxonomic level. We had 3 domains, archaea, bacteria, eukarya, and now we're looking at the level right underneath that. And there's some of them that I just can't pronounce, so I'm sorry about that. Excavata, I can pronounce Excavata. Excavata are going to be a whole bunch of what we would call protists. There used to be a whole kingdom called Protista, but since we've been sort of siphoning off protists into different supergroups, now most of them end up in this group called Excavata. It's called Excavata because it's got this feeding groove to it. It's got a funnel. All excavates have this. A lot of these organisms with feeding grooves have flagellar cilia, whipping the environment in order to push food into that groove. Once it gets to the bottom of the groove, it's going to undergo a process called phagocytosis. The food is. Phagocytosis is where you're going to form a unicellular organism forms a vesicle around a food particle. I believe we talked about this previously when we talked about endocytosis and exocytosis in Bio 101. I hope you remember that. Because the idea is you've got a membrane, actin filaments along that membrane begin to contract, and it folds inward to create a bubble. That beginning vesicle allows the food to land. It gets surrounded by the rest of the membrane, which pinces off and brings it into the cell. That then will be digested by a lysosome. Digestive enzymes will bind to the vesicle, and you get your food from that. So I guess phagocytosis is food cell movement. Phagocytosis provided the mechanism by which endosymbiosis could occur. Endosymbiosis being where 2 organisms came together for mutual benefit. One of them took the other into its cytoplasm but didn't digest it. I think we've talked about endosymbiosis in here, haven't we? I know we talked about it in 101. Talk about lots and lots of many rounds of endosymbiosis. Speaking of lots of rounds of endosymbiosis, the Euglenoids. Euglenoids are a major group of protists that are characterized by an interlocking protein layer under their plasma membrane. That makes them very flexible. You've probably heard of a Euglena. Euglena is a heterotrophic, well, it's a photoautotrophic heterotroph. It's green, it takes in the sun's energy, but it does not, but it's also an active hunter. The big thing that characterizes euglenoids, though, is flexibility. Because not all euglenoids are Euglena. Most don't have photosynthetic capabilities at all. They do, though, have what are known as plastids. You guys know one plastid very well. What is the plastid you know? The chloroplast. And the chloroplast is there to synthesize organic molecules. That's what most plastids do, synthesize molecules. The chloroplast happens to do it using light. Others do it using different types of energy. A kinetoplastid is a very, 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 very large mitochondria, and it contains a huge amount of DNA. Typically, you're going to see these at the base of a flagella. So that was the excavates. The excavates and the euglenoids. Another major supergroup, and this one should be familiar to you guys, is Streptophyta. You may not know the name, but what it means is the land plants and all their relatives. You've seen them. You interact with them on a daily basis. You eat them. Streptophyta is all the land plants, but it also includes all the algae. And that's what I want to focus on here is the algae. Today's a big algae. Algae live everywhere. They're photosynthetic. You've heard of them before. You've seen them everywhere. When conditions are right, the algae are going to bloom. If there are a lot of nitrates present, you're going to see a large influx of algae. Like I said in the last lecture, this tends to happen at the mouth of rivers. Why? Why at the mouth of rivers? It's not where water interacts with water. It's not where it's the most still. What's flowing into the river? What? Fertilizer. We use an insane amount of nitrate to fertilize our soils. We cannot produce the crops we grow using the conditions we do now. Right now, crops are being grown, typically the same crop grown year after year after year, and that's going to require a whole lot of influx of nitrate. So how do you add nitrate to a soil? You spray it. All right, what happens when it rains? It runs off. Where does it run off to? The only place it can, to the creeks. The creeks flow into the rivers. The rivers eventually flow into the ocean. So you end up with a lot of nitrate at the mouth of a river. Heck, there's a really big river in the United States. What is it? Mississippi. So literally, there's a continental divide, like right in West Virginia, where all of the everything to the west of the divide flows into the Mississippi. Everything to the east flows into the Chesapeake. All of the fertilizer from all between West Virginia and the Mississippi River flows into the Mississippi. From the Rocky Mountains to the Mississippi flows into the Mississippi. All that fertilizer. And we're just dumping it into the Gulf of Mexico. So you have a lot of nitrates there. And as such, you have a lot of algal blooms there. Algal blooms can produce a lot of toxins. If you touch them, you can get sick. If you breathe them, you can get sick. But as far as more important to the ecosystem is their impact on gases and in the region. Algae are photosynthetic. What does that mean? I'm going to have to walk you. I want to walk you through the step by step so you can see my point. What does it mean to be photosynthetic? They need sunlight, and what do they make with that? Energy. How? Okay, well, they take the sunlight and they create glucose, right? They use carbon dioxide. So they're pulling carbon dioxide out of the atmosphere and releasing what? Oxygen. So far, so good. Life's happy. We're producing oxygen. We should all be thankful for that, right? Algae, we just said, produce half of all the oxygen on the planet. Algal blooms can only help with that, right? I mean, you add a lot of carbon dioxide to them and they produce oxygen. The deal is, though, when does that happen? That happens during photosystem, photosystem 2, which happens before photosystem 1. Photosystem 2, you're taking, you're releasing oxygen. When does that occur? Photosystem 1 happens when? When where? When there's water? Think more. You're right. You do need water. You need the sun, right? When's the sun out? Not trying to trick you. During the day. What happens at night? At night, no sun, but the plant still needs energy. It's still got mitochondria. How does it get its energy? At night. No, not from water. Using oxygen. Cellular respiration. So at night, those plants stop producing oxygen, and instead they start consuming it like crazy in order to stay alive. So they take the oxygen from the water. That leads to anoxic zones where there's no oxygen present. Well, where there's no oxygen, you're not going to have fish. Fish need, in case you didn't know, I keep forgetting. Okay, so Bio 102, you'll probably know this. Fish need oxygen to survive. They don't come to the surface and breathe it. They drink. They breathe dissolved oxygen by pushing it across their gills. So at night, the oxygen levels drop dramatically and the fish die. That leads to large die-offs. It also leads to fish and all of their associated organic components having to be broken down. The bacteria that live in an anoxic zone are anaerobic, and oftentimes they're going to produce methane. So you've got oxygen levels dropping during the night, methane being released, which leads to increased temperatures of the water. Increased water temperatures can't hold oxygen as well. So right now there are giant dead zones in the ocean where nothing can live because of the algae. You can't, fish and can't breathe in it. You can't swim in it. Last summer, Florida was utterly covered in a toxic bloom for months. Every little canal, every byway, it was everywhere, at least on the East Coast. I'm sorry, the West Coast of California, Florida, sorry. Algal blooms, though they may seem like they could help with the environment, are actually environmentally detrimental. One type of algae are red algae. They're red. They're not using that 520 wavelength in order to produce sugar. We use red algae extract for producing agar and agarose. You may have seen, it's sort of like this jello-like substance that you grow bacteria on. Red algae and even green algae to an extent are very complex because they've had multiple rounds of endosymbiosis. This means that you have primary endosymbiosis. What happened with the mitochondria and the chloroplast? How many bilayers do mitochondria and chloroplasts have? 2 bilayers. Now imagine this. An organism had endosymbiosis occur. So it's already got an organelle inside that's got 2 bilayers. That organism is then taken up by another organism. So now you've got, geez, let's count the So we have, here's our organelle inside of another cell. That one is taken in by another cell. Another round of endosymbiosis occurs. So now we have 1, 2, 3, 4 bilayers. That's secondary endosymbiosis. You could have tertiary endosymbiosis. 6 bilayers happening again and again and again. And what this leads to is organelles with multiple membranes. And having many different membranes means you can have lots of compartmentalization. Makes them efficient at whatever job they've got. It also leads to a lot of horizontal gene transfer, moving DNA from one of the organisms to another. They're in such close contact. In you have mitochondria surviving. Everybody good on that idea? Is the DNA in the mitochondria your DNA? Yes? Yeah. Most people, I think, would agree that the DNA inside your mitochondria makes you. These organisms have had the DNA in their various organelles moving from one level to the next within the different organelles. They exchange it back and forth. So we've gone from having not just one type of supergroups DNA in there, but lots of different types of supergroups. And they all get mixed up. Heck, you have DNA in you that you've picked up from viruses. Every time you catch a cold, there's a chance that you will take some of the genes from that virus and integrate it into your body. When you have a reoccurrence of a virus at some point later in your life, this happens with shingles with some people. You guys never had it because you all got vaccinated for chickenpox when you were younger. But shingles, it would tend to hide in the cells because the way it hides is it integrated its DNA into your genome. You have little time bomb cells inside of you that are programmed to go off later in your life. It's Surprise. So every type of streptophyte out there, every land plant has plastids with these multiple levels, with their own DNA in them. All right, I'm going to go a little bit less serious then with the, because streptophyte is going to be our big topic for the next lecture. But I want to talk about Alveolata. Alveolata have sacs. That's my thing with them, sacs. Little pockets. Pockets are, in fact, in case you didn't know, the coolest thing ever. If you've ever worn any outfit that lacks pockets, life sucks. I always, in case you haven't noticed, I always have a pocket right here. And I inevitably am like, all right, I'm going to stick my pen in here. I'm going to stick, like, random stuff in here. People were making fun of me at the last professional development because, like, I was eating candy, and then I would take the wrapper and put it in my pocket. Because I was like, well, I got to get rid of it, and I don't want it to sit on the table in front of me. Pockets are great for you. Pockets are also great for other organisms. They have a purpose. They might be used for flotation, defense, or increased reproduction. They work for flotation. If you've got pockets, hypothetically, you're a cell. Not you. You're a cell. You got these little pockets. You can fill those pockets with gas, and that makes you float. Your waste gases can go into those pockets, and you can float up. You release them as bubbles, and you go down. Defense. You literally have poison in your pocket. Some of you guys might carry, I don't know, mace around or something, right? You keep it in your pocket, and then when you need it, you pull it out, and you can mess up whoever's attacking you. Why? You got pockets. Increased reproduction. This doesn't necessarily go with us, but hypothetically, these guys go around with sex cells in their pockets, and they pull them out and throw them like glitter. It increases reproduction. Another group, Stramenopila, they have hairs. That hair serves to increase surface area. Because the more this flagella has surface area, the more water it can move, the more it can push. It's just like, I don't know, have you guys ever been on a canoe? Okay, you know what an oar is? Okay, whew, okay. If you have an oar, you stick it in the water, and you push the oar, and it moves water. We're all good on that part, right? The difference between Stramenopila flagella with hairs and one without hairs is the difference between an oar that's got a flat bottom to it and a broomstick. If you use a broomstick to move water, you can move the water. You can, in fact, move the boat with a broomstick by going like this really fast. But if you have an oar that's got this wider surface area, it takes a lot less energy to move it. Probably the most famous Stramenopila is Phytophthora infestans, which is a fungus-like Stramenopila. It was responsible for the Irish potato famine. This one stupid protist wiped out most of the potato crop in Ireland. It still leads to billions of dollars of losses every year in potato. The way it moves is it swims. If it's moist out, if there's water, and you've got a Phytophthora infestans protist or group of protists living on one plant, let's say it rains. Now they can swim from one plant to the next. If the field is saturated, they can grow all through the field, wiping out crops. You have your brown algae, kelp. Kelp is a type of Stramenopila. Kelp can grow up to 100 meters tall. Yet they are still just protists. Rhizarians, different group. Rhizarians have spikes made of cytoplasm. Rhizarians are one of the few microorganisms that you can actually get fossils of. We use rhizarians for dating because they incorporate carbon into their cytoskeleton, which is then saved. Our amoebozoans, go figure, they're the types that use pseudopods, like amoeba. Slime molds are amoebozoans. Slime molds are one of my favorite protists, if not my favorite protist. If you leave beer out for any amount of time that's longer than like a few hours, slime molds will migrate toward it. They love this stuff. They're unicellular organisms that can then switch over from being unicellular to multicellular. And there's almost a link between them and multicellularity. In fact, I want to show you guys a video shortly on slime molds because they just are awesome. I want a pet slime mold. If you guys find slime mold, just bring it, and I'll put it in a petri plate. We'll feed it some oats. They'll be so happy here. You can teach them to do tricks. I can absolutely keep slime mold alive. I cannot keep a fish alive to save my life. They're hard to find. I mean, I could leave out beer, but seriously, it's a waste. So, there are priorities here. There are priorities. I don't want a slime mold that much. But if you guys could find a slime mold, that'd be awesome. We'll do some experiments with them. Slime molds are cool because slime molds actually set up farms. They literally build little corrals where they grow bacteria. They send the bacteria food in order to grow the bacteria as make the bacteria grow stronger, make them grow in more numbers, and then when it gets to a certain point, the slime mold covers over most of the bacteria, devours them, and then expands back out, and then starts feeding the bacteria that are left again. So you sort of, just like we farm things. And we got our episthecons. These are the ones that are closest to us, and this is how we're going to start segueing into multicellularity. Nucleria are going to move off to be fungus. This is a very simple protist. It looks a lot like a fungus. It's one of those fungus-like protists, but it's missing a cell wall. That's its big issue. The rest of the episthecons, though, one of the examples is the Choanoflagellate. And I want to talk about the Choanoflagellate because it's got some adaptations that may have indicated this is the first one that went on to multicellularity. They have cellular adhesion genes and extracellular communication genes. They can talk to each other, and they can hold on to each other. You can go from a single structure to a multiple protist structure. They start connecting with each other, they start talking, they start communicating, and soon you have multicellularity. This is the inside of a sponge, a multicellular organism. And you'll notice that the cells look surprisingly like those choanoflagellates. In fact, they're called choanocyte cells. There's a debate, there was debate, it's no debate anymore, on whether this is direct evolution of the choanoflagellate to the sponge or convergent evolution. But based on the molecular sequences, it absolutely looks like this is direct evolution. They have these same sequences. All right.